So this is cell-mediated immunity. Remember that cell-mediated immunity is all involved with T lymphocytes, T cells. Now, the ultimate goal of this is to kill infected tissue cells. And killing tissue cells is an expensive process for our body. We spend a lot of time growing cells, we spend a lot of energy maintaining them, and we need the function of those cells in order to maintain homeostasis and health. So it's not in our best interest to just be randomly killing cells on the suspected of them not being right. So there are the cells that kill those cells. They're called cytotoxic T cells. And there are cells that whose job it is to check that the cytotoxic T cells are in fact doing the correct thing and not getting out of hand. So in order to activate a T cell, what they basically need is they need to be co-stimulated. They need to be stimulated twice. And when the two stimulations come, when the two signals come, then they get activated. So there are receptors that bind to that antigen fragment presented in the MHC complexes. So these are processed antigens. There's a couple of different types of receptors. One is called CD4, the other one's called CD8. I'm not that worried about it right now. But that's not enough. That first signal just says, okay, we've identified a problem. The second signal is called a co-stimulation. And it's basically chemicals, cytokines or various things, that are released by cells called helper T cells that basically give permission. They say, yes, we recognize that this is a problem, and therefore it needs to be dealt with. Go ahead and kill. It's almost like the cytotoxic T cells or the SWAT team that are about to blow up a house in a neighborhood that they suspect has terrorists in it, and the helper T's, the second signal, comes from the police captain that says, yes, do it. I mean, if you watch Flashpoint, there's always the guy that's going to take the shot, and then the guy that says, yes, take the shot. So if the thing has a CD4, it will develop into what's called a helper T cell. And their job is to recognize the fragments and with just in the antigen presenting cell. So that would be like a macrophage that, or a neutrophil or a dendritic cell that has ingested the antigen pathogen. And then when it becomes activated by the antigen presenting cell, it undergoes clonal selection. It makes a lot of active helper T cells and memory helper T cells to that specific antigen, right? And they get involved with secreting various extracellular substances, cytokines. Cytokine moves cells, right? And they help with this co-stimulation of the killer T's. Now the memory T cells aren't active in this round, in this infection. They are saved for later. They're long-lived cells, and they quickly proliferate and do it again if the antigen appears again. And chances are good that if the antigen is in your environment once, it will be in your environment again. So what happens here is the antigen presenting cell the macrophage has eaten the pathogen, presented chunks of the antigen on the surface, and it's MHC2s. The inactive helper T cell binds with it and then is co-stimulated by the fact that this is an antigen-presenting cell. It won't bind with a regular cell and then be co-activated. Only the antigen-presenting cells will do both. So it stimulates once and then is co-stimulated a second time. That becomes an activated helper T cell, and then that activated starts the cloning. And it basically creates two populations: active helper Ts, which are going to be releasing the cytokines and be co-stimulating the other cells, and memory helper Ts that stick around for the next infection. Now, if the receptor is something called a CD8, then the T cells develop into what are called cytotoxic T cells, or more commonly referred to as killer Ts. Now, they bind with antigens, and they recognize antigens combined with the MHC1s. These are the common MHCs. These are the ones that are in all the body cells except for red blood cells. Now, what they need, once they've been partially stimulated by the MHC1s with the antigen, is they need to be co-stimulated with MHC2s and by the helper Ts. Once they're activated, they undergo clonal selection, and it makes two types of cells: the active cells that attack body cells that have that MHC1 antigen combination, and memory killer T cells that don't attack, but they wait for that antigen to appear again. So they wait for a second round of that infection, encountering that infection again in the environment. So this is why you catch something at work, you fight it, you get sick, and in the process of getting better, you give that disease to your spouse. Well, they don't get to give it back to you because now you have memory cells, you've recovered from it. So it works like this: the infected body cell presents its MHC1s here, comes to the inactive killer T, which will attach to it, and then is co-stimulated by the helper T cell that's been activated by the same antigen. So it's almost like this cell goes, this one pisses me off. Should I kill it? Yes. Then he does it. So the activated, then, first thing it does is it clones. It makes a whole bunch of active T cells, killer Ts, and then makes a bunch of memory killer Ts for later. Are we seeing a pattern in this? Memory is an important thing. So then the cytotoxic T cells start to kill. They kill the infected target cells, the cells that are present in the MHC1s with the antigen in it. Now, they don't kill any cells, they just kill cells with that specific antigen in its MHC1 antigen fragment. They do it just like natural killer cells. Natural killer cells are a type of lymphocyte that do anything that's got an MHC1 that's wrong. This is much more specific. So they do it with perforin and granulozymes, so cytolysis and apoptosis. When a cell is cancerous, it presents antigens that are not quite self, and we will recognize those and do the exact same thing. And we'll kill them. So So when the microbes in the infected body cell, what happens is the activated T cell releases the granulozymes and the lysozymes, and it basically kills this cell, releasing all the microbes into the interstitial fluid where they get eaten by macrophages. It's the same thing here. If the perforin will perforate holes, release these things, and they get eaten by macrophages. In both cases, the cytotoxic T cells release the microbes from inside the infected body cell. Natural killer cells will do the exact same thing. They release these microbes into the interstitial fluid where the microbes are dealt with by the phagocytes. The phagocytes actually eliminate the microbes. The killer T cells don't, or the natural killer cells don't. They get them out of the house where these cops arrest them. It's a bad case. The trick behind this is this idea of co-stimulation, and the important thing to remember is that the cytotoxic T cells do not, of their own, kill any microbe. They only liberate the microbe into the interstitial fluid, where the killing is done by the phagocytes. Next will be antibody-mediated immunity.